Hello everyone, I am Dr. Richa from Physiology Open. Today we will talk about drug distribution, continuing with our Pharmacokinetics lecture series. So what is this drug distribution? As the name indicates, distribution. As we give the drug and it is absorbed into the plasma, from there it distributes into another location. So that is drug distribution. For example, suppose if this is the person and this person will have all the tissues and we are injecting the drug in the plasma. If we are giving other routes also, ultimately it is going to enter into the plasma. Now from the plasma, it will go into various tissues. Say suppose this is the plasma and finally it has reached there. Now it will go out of the plasma into the various tissues. Okay, and it will move until and unless it has become equilibrated, that is the concentration it is same everywhere. So that is the concept of distribution. If especially it is a lipid soluble drug, then it will based on this concentration gradient, it will move and equilibrate with other tissues. Now finally there will be a plasma concentration which will be achieved after equilibrium, right? So this plasma concentration helps in determining the volume of the distribution. That means in how much volume the blood has gone into the various tissues and distributed, right? So simple formula we use, that plasma concentration, say suppose if it is X milligram per deciliter, then total amount of the drug is equal to the volume into concentration. This is simple. Mass is equal to volume into concentration. So that formula we use. So amount will be equal to, in this case, volume will be volume of distribution, and this amount divided by the plasma concentration will give us the volume of the distribution. So this amount we will know how much amount we have given. Suppose if we have injected the drug, so then because of 100% bioavailability, whole amount will go into the plasma, isn't it? And from there it is going to redistribute. So in injected case, this amount we will know. If it is other routes, then whole amount will not be absorbed. Okay, so that is why we are taking the injected one. So amount divided by the plasma concentration will give us the volume of the distribution. So what is the unit of this volume of distribution? It will be in liters, the volume in which the drug has distributed. Sometimes it will be given as liters per kg. In your books you will see why it is given per kg. Per kg we take it the total body weight, total volume what we have got from here. We take the total body weight and divide this by the total body weight. So it is normalized with different body sizes. So when we have to compare the volume of distribution in different people, then we divide it by the body weight so as to normalize across different people. So that was the simple basic concept. But remember that though here we have explained it so easily that drug will go everywhere, ultimately it will equilibrate, but that doesn't happen. This concept assumes that body is a homogeneous compartment. That means everywhere it is behaving same. But actually it is not happening. Actually when the drug moves, it may go more in certain organs, and because of that the plasma concentration is going to decrease further. Say suppose there is a tissue, and what happens if this is crossing, and there are certain tissue proteins, and as it crosses, this drug binds with this tissue proteins. So the concentration here in this tissue, that will not be equilibrated faster with the plasma concentration, isn't it? So as it is going, it is mopped up by this tissue protein. So plasma concentration is not getting equilibrated. Hence more and more drug is going to cross into the tissues, right? Similarly, there may be certain tissues to which the drug goes much more compared to other tissues. So again, the plasma, the concentration in these tissues will not be same. You can say that the amount which goes into these tissues may not be same. But the concept here is what we want to know, why we want to study here, that when we are giving the drug and it is going into the various tissues, ultimately the plasma concentration which is achieved depends on that how much of this drug is going into the tissues. Suppose it is going much more to all the tissues, what will happen? The plasma concentration which will be achieved will be much lesser, isn't it? So what we will get, that plasma concentration, suppose we determine the sample, take out the sample after equilibration, and the plasma concentration we get, it is much less. That means the volume of distribution of the drug is much more. It is inversely related, isn't it? So why do we want to know this, that volume of distribution is more? Because it is important in calculating the dose. Yes, we want the plasma concentration to be maintained in a therapeutic range. Otherwise, if it is too much distributed to other tissues, then we may need to increase the dose, right? So that is why for a particular drug, we need to know the volume of the distribution, and especially it is required to calculate the loading dose. Why? What happens, that loading dose will help us maintaining the plasma concentration. So we give a higher dose so that whatever drug has to go into different tissues, that is known as sequestration. Suppose it is getting sequestrated there, and all these sites where this drug can bind all are filled up, then no further drug can bind to those proteins, and all the sites will be filled up. So that will help us maintain the plasma concentration, because now the drug will not go much more to those places. Okay, so loading dose is the one which is calculated based on this volume of distribution. Second thing is that there might be some drugs which may have more toxicity because they are getting sequestrated in certain tissues. For example, tetracyclines. Tetracyclines go much more in bones and teeth and bind there. So we get tetracycline toxicity in these places. Third is the hemodialysis concept. See, if a drug is going and binding to certain tissues and much less of it is available in the plasma, and if toxicity occurs, then when we are doing hemodialysis, in hemodialysis what is happening? We are taking the concept of drug diffusion. Okay, so the drug will diffuse whatever is present in the plasma from the because this is going to pass to a filter, right? And that diffusion will happen for the drug which is present in the plasma, not the drug which is bound to the various tissues. So hemodialysis becomes difficult for the toxicity of that particular drug, right? So this is the fundamental concept of drug distribution. Remember that this is a hypothetical concept because we are assuming that body is a homogeneous compartment, single compartment is there. There is a multi-compartment model also, like Goodman-Gilman has talked about a multi-compartment model, where it has talked that there may be some tissues in which the drug goes much faster. Okay, and these are the places where the perfusion is much more, like brain, heart, kidney, and there may be other compartments where the perfusion is lesser, so there will be slow movement of the drug to those compartments. So first one is known as the first compartment, and then we have the final compartment, right? But this one is the most easy one to understand, that what is volume of distribution. Now, what are the factors which will affect this volume of distribution? Well, we have the body characteristics which will affect the volume of distribution, and we also have the drug characteristics which affect this volume of distribution. Now, body characteristics, what effect they have on the drug? That is what is pharmacokinetics, right? Body characteristics which have effect on the drug. So that is why we are studying this in distribution. But remember, distribution is also determined by the drug characteristics. Okay. Now, we will see many body characteristics. What are these? Obese versus lean. If a person is obese, obviously the drug distribution will be much more in obese people, because there is an extra tissue is there, fat is there, so drug is going to go into the fat as well, right? Edema versus non-edematous person. If a person is edematous, that means the fluid is going out of the vascular compartment and here it is accumulating, and because of that the body is retaining more water from the kidneys. This is fundamental physiology. Whenever there is edema, the pressure in the vessels, it is going down, so kidney activates renin-angiotensin system and there is more retention of the water, so vascular compartment will be filled. But this extravascular compartment, the interstitial space, there will be accumulation of water, and obviously now the drug has to equilibrate in more compartment. So that means edematous person will have more volume of distribution. Again, what is the importance? Yes, dose is going to change, right? Then adult versus child. What is the difference? There are two kind of different volume of distribution may happen. One, based on whether the drug primarily remains in the blood vessel. That is the property of the drug. If such kind of drug is there which primarily remains in the blood vessels, in the fluid it is remaining, then volume of distribution will be more in children. Why? Because total body water is more in children. Okay, it will be more, sorry. Okay, on the other hand, if it is going to the tissues, then the size of the tissues is smaller in children, then the volume of distribution will be more in case of adults, if the drug is going preferably to the tissues, right? Because the number of cells will be more in case of the adults. Then coming to old versus young, in whom the drug distribution will be more? In old it will be more, because in old adipose tissue is much more. Okay, and this drug distribution is much more in adipose tissue. Generally, for most of the drugs, it is much more for adipose tissue. So, adipose tissue more means for old volume of distribution will be more, right? Then the tissue perfusion also determines the volume of distribution. Say, suppose in condition like shock, what is going to happen? That blood flow to various tissues is going to decrease. That means the drug will not go to the various tissues only, it will not cross to the tissues. That means it is going to remain in the vascular compartment. So in this case, volume of distribution is going to decrease. That means whatever drug you have injected, it is going to remain in the plasma. That means the same dose of the drug, now it is going to create a higher volume, higher plasma concentration of the drug. Right? Then cirrhosis, suppose it is there. Again, the vascularity of the liver is decreased, so less drug is going to go into the liver. Okay, so these are the body characteristics which affect the volume of distribution. Then, what are the drug characteristics? Molecular size. If the size of the drug is more, then it is not going to cross the vessel. There will be very slow crossing of the vessel. So, volume of distribution will be lesser for higher molecular weight. Lipid solubility, if the drug is more lipid soluble, it is going to cross much faster. And we have seen in the video on absorption of the drug that how pH affects the absorption as well, that it leads to ionized and non-ionized components depending on the pH, right? So pH will determine the lipid solubility as well. That means say suppose acidic drug and acidic pH, that means it will be more in the non-ionized form, and that means it is going to increase the lipid solubility. So that is going to increase the volume of distribution because the drug is going to cross more. Plasma protein binding of the drug. Remember that acidic drugs, A for acidic, A for albumin. Acidic drugs bind more to albumin and basic drugs bind more to alpha-1 glycoprotein. However, most of the drugs which we are using are acidic drugs. Basic drugs are the ones generally which are derived from the plants. Okay, so most of the drugs used are acidic drugs. Anyway, so plasma protein binding, how it will affect the volume of distribution? Say suppose this is a drug which is bound to the plasma protein. Now, this drug will not be able to cross the vessel membrane, isn't it? Because albumin or any plasma protein does not simply cross any cell membrane, right? So that means this drug is going to remain in the vascular compartment. So any drug which is more bound to plasma proteins, for them volume of distribution is lesser. On the other hand, there might be drug which is having more tissue protein binding, like the one which we studied here, that when it is going, it is binding to tissue proteins and getting sequestrated there. So in that case, volume of distribution of the drug will be more. So that was about the distribution of the drug, right? But when we talk about this plasma protein binding, we need to know certain concepts as well. So if there is a drug which binds with plasma protein more, then there are certain characteristics which we are going to see. One, the drug is going to become long acting. Why? Because this drug, since it is not able to cross the plasma membrane, the cell membrane easily, It is not available for metabolism. It is not available for excretion because it just cannot simply enter into the liver and enter into the nephrons. It will not be able to filter into the nephrons. We know that plasma proteins don't get filtered. So that is why this drug which is bound to the plasma protein will also not get filtered. Hence it is not available for metabolism, not available for excretion. So it becomes long acting. So fundamentally, this acts as a storage, temporary storage of the drug. Plus, this is a reversible binding. It is not like permanent. So drug molecule can detach, attach back, detach, attach back. Whatever drug is in the free form, because not everything will bind. Whatever drug is in the free form, it will get metabolized or excreted, right? Then this attached form will be released depending on how this drug is getting metabolized. Okay, so it is acting as a temporary storage form. Then again, hemodialysis is not effective for a drug which is bound to the plasma proteins. It has a very high binding to the plasma proteins. Okay, because again, hemodialysis we saw that it is about the diffusion. If the drug is bound to the protein, then it cannot easily diffuse, right? Then there might be displacement reactions. Say suppose there is a drug which has bound to the plasma protein and now another drug is also there which can also bind to the plasma protein, and say suppose that this drug has high affinity for the plasma protein. So I told you this is reversible binding. It will attach, detach, right? So when this drug will come and this is detaching, this will attach to the plasma protein, and since it is high affinity, it will not easily detach. So ultimately we will get this particular drug, the drug with the low affinity for the plasma protein, it will be released into the blood as free form. Now that means that plasma concentration of this drug is going to increase, and this can even lead to toxicity. Okay, so that is one thing, that plasma concentration is going to increase. But remember that when the plasma concentration increases in the free form, then it will be available for metabolism also and excretion also. So very short duration, yes, the free form is going to increase, but later on it will be available for metabolism, excretion, and again its concentration, its free form concentration is going to go down, right? But still this knowledge of bound form and free form is important, because the drug is active only in the free form, not in the bound form. It cannot go into the cells, it cannot act, right? So for example, antibiotics, when we are seeing the concentration of the antibiotics, the bound form, if we say that the total concentration is bound form plus free form, then it is not the effective concentration, because for acting it should be free, right? So we have to be mindful of that. And also of the hormones, say suppose the thyroid function test we are doing, then we get the free form as well as the bound form, and we are interested more in the free form of the hormone, because that is the hormone which is the active hormone, right? And there are various conditions in which if this plasma protein concentration increases and this drug can bind with the plasma protein, what is going to happen? Bound form is going to increase, because the concentration is increasing, the drug is going to bind with the plasma proteins, right? And this is going to decrease the free form concentration which is active. Now coming to redistribution, the final concept. What is this redistribution? Certain drugs which are highly lipid soluble, for example, one drug is thiopentone. What happens that as we inject, first it goes to certain highly perfused areas like brain, heart, kidney. Okay. Then slowly it goes to areas which are not perfused that much. For example, muscle and fat. Okay. So first it goes to these areas, right? Its plasma concentration as it decreases and these areas get like filled with this. Then it starts going into the lesser perfused areas. Now, when it starts going into the lesser perfused areas, its plasma concentration falls further. Okay, it falls further. Now these areas, because of the first distribution, they have more concentration. So as the plasma concentration falls, the drug starts moving out of these areas, the initial area. So that is known as redistribution of the drug. and this may be responsible for termination of action of the drug. For example, if it is acting on the neurons, if it is acting on the brain, now the drug is getting redistributed, so no more it will be able to act on these cells. So redistribution may be one mechanism of the termination of action of the drug. So that was about the concept of distribution of the drug. With this, we have completed the first chapter of pharmacokinetics of KDT. We will move on to the next chapter in the next video. Thanks for watching the video. If you liked it, do press the like button, do share the video with your friends, and don't forget to subscribe to the channel Physiology Open.